Great, yeah, thanks so much. So I'm in the chemistry department. My lab does biotechnology and synthetic biology approaches really geared ultimately at trying to find both diagnostics, but more importantly, cures for human diseases. And what I want to talk about today is related to a technology you may have heard of called the CRISPR-Cas system, which is a bacterial protein that allows one to go in and change the code of DNA anywhere you want based off of the code of that site of DNA. And this evolved as a bacterial immune defense system. Now it's being deployed as a therapeutic, and for the first time, one can go into a patient, identify the site of a DNA change that's causing that person's disease, and correct that site using this CRISPR-Cas system. It's transforming the way we think about developing therapies, and it's happening right now. So if you happen to really care about one of those disease areas that are curative using that approach, this is going to really change the way we think about that disease in the coming years. However, there's a limited scope of when you can do that. Because these are large bacterial proteins, it makes them difficult to apply in humans because you're going to have an immune response to those therapeutics. So my lab has been really thinking about how we can overcome those limitations of those natural systems, and what we've come up with is a humanized form of this CRISPR-Cas system. So you don't have a gene editing tool within your genome. Humans never evolved them. However, we have lots of other interesting building blocks, molecules within us, and what my lab has developed is a method that we can engineer them together into a humanized gene editing tool, so that when we can ultimately deploy them as therapeutics, we can avoid those immune backlash issues and go after not just kind of the monogenic, rare diseases that are currently being exploited therapeutically, but the really big issues like heart disease, stroke, neurodegeneration, wound recovery, and the gastrointestinal tract, as well as other areas of the body, and use these gene editing tools to solve them. So one of the reasons I started my lab at the University of Chicago is I knew my motivation is technology development, and I wanted to do that in a clinically relevant sense, and being next to the hospital and talking to clinicians really fuels that within my lab. So yeah, working with Gene and now Dr. Rubin really motivates my lab, and with platform technologies like this where we can move in many different directions, having clinicians to tell us what the important problems are and where we should be going with that technology really helps motivate the lab, but also make sure we're spending our time moving in the right directions. And certainly support from CURF allows us to think about how we can apply these to diseases that are relevant to all of you, and also important within the clinic. So thank you, and I'm happy to answer questions.
Thank you.